The third method is Plaque assay. Plaque assay is commonly used to determine viral titer. Why determine viral titer is so important? For example, we want to make vaccine. We should cultivate virus to enough amount, so we should measure what's the titer of the virus. And another thing is when we use medicine for treatment, we would like to know whether the patient's viral titer decrease. If the virus decrease, indicating the medicine can work. This is why we should use Plaque assay to know viral titers.
A confluent monolayer of host cells is infected with the virus at varying dilutions and covered with a semi-solid medium, such as agar or carboxymethyl cellulose, to prevent the virus infection from spreading indiscriminately. A viral plaque is formed when a virus infects a cell with the fixed cell monolayer. The virus-infected cell will lyse and spread the infection to adjacent cells where the infection to lyse cycle is repeated. The infected cell area will create a plaque which can be seen visually or use an optical microscope. Plaque formation can take 3 to 14 days, depending on the virus being analyzed. Plaques are generally counted manually, and the results are used to calculate the number of plaque-forming units per sample unit volume. A PFU per ml result represents the number of infected particles within the sample and is based on the assumption that each plaque formed is representative of one infected virus particle. Seed cells into 6-well plate and incubate at 37 degrees Celsius for 24 hours. The cells should be 90% to 100% confluent. The first step is to dilute virus. Virus is sold at 37 degrees Celsius in water bath. Fetal bovine serum FBS may affect viral infection, so we use the culture medium without FBS to dilute virus. Each tube contains 1080 microliter culture medium without FBS. Next, add 120 microliter of virus into the first tube. Mix well and take 120 microliter of virus into next tube. Make 10 times serial dilutions with the following tubes. To infect cells with virus, discard medium from each well and wash cells with PBS. Then add 500 microliter diluted viruses into each well. Incubate viruses and cells in 37 degrees Celsius for 1 hour. Shake the plates every 15 minutes. The agarose solution contains 0.3% agarose and culture medium with 2% FBS. An autoclaved 3% agarose solution and culture medium are pre-warmed at 45 degrees Celsius. Add 4 ml 3% agarose solution into 36 ml culture medium. Mix well, then keep at 42 degrees Celsius. Discard medium from each well. And wash cells with 500 microliter PBS. Add 3 ml 0.3% agarose solution into each well. Keep the plates at room temperature until agarose overlay solidify. Then incubate the plates at 37 degrees Celsius for 60 to 84 hours. Fix the cell with 3.6% formaldehyde solution at room temperature for 1 hour to 24 hours. Remove formaldehyde solution, then wash out agarose overlay. Stain cells by 0.5% crystal violet at room temperature for 1 minute. Rinse each well with tap water and remove plaques.
Finally, I'd like to talk about the last part of today's lecture regarding treatment and prevention. For treatment, there is a medicine on the way to clinical trial called Plaque. The rationale is that this chemical can block the receptor binding sites, so the virus cannot enter to the cells for replication. And we believe such kind of design will become more and more popular, so we got a hope to have the medicine to treat poliovirus or other enterovirus infection in the future. Another way for prevention is to develop vaccines. There is a very successful story is for poliovirus vaccine, Salk and Sabin. Salk is inactivated vaccine and Sabin is live vaccine. Live vaccine got a risk to mutate back to the wild type and cause disease, so relatively Salk is more safe. This is Dr. Salk and this is Dr. Sabin. Their story, their contribution is kind of very wonderful in this field. And in the middle, there is a figure, is the record that children got infected and then paralysis before. It's more than 5,000 years ago. So this disease existed for so many years until last century. Dr. Salk and Sabin solved the problem, saved so many lives, and prevent children to suffer paralysis. They saved so many lives before and become a very wonderful story in the field of biomedical science. After introducing the basic knowledge of this virus, how it replicates, how it was diagnosed, and also show you how to do the laboratory diagnosis, how to do the experiments including cell culture, RNA extraction, Plaque assay, and RT-PCR. Then I hope you now got more idea about enteroviruses, which is a very important pathogen. And even though we have poliovirus vaccine, but we don't have vaccines for other enteroviruses, and we don't have drug so far. So I hope you recognize this is an important pathogen, and after knowing this, you will get some motivation to do some research or do some work and contribute to this field. Finally, I'd like to thank you all of you for your attention to listen to this lecture. I do hope you learn something and can have a better understanding for enterovirus.